Chapter 4 is all about cells. So in this chapter, we're going to talk about cells in general, but we're also going to talk about the two different types of cells. We're going to talk about parts of cells, and we're going to talk about how you move stuff in and out of cells. So first, we're going to just talk about cells. So all organisms are made out of cells. Cells are the smallest part of a multicellular organism like yourself. And the cell is the smallest unit of life that can function. So we've got multicellular organisms like this guy here, or fungi, or plants, or elephants. But we also have single cell organisms. So I've got a little show and tell. You'll see these in other lectures as well. I've got stuffed microbes. So this is a singular cellular organism. Obviously, it's a little bit bigger than normal, but this is a paramecium, and it's a single cell. It's an organism that can live on its own, and it's one cell big. That's all you have to be alive. There are other things that you need to be able to be alive, like you need to be able to process food and reproduce, and that's exactly what a cell is. It's the smallest unit of life that can function on its own and perform the necessary functions like reproduction, getting food, all those sort of things. Now, cells come in different sizes, and so you can actually see individual cells. Now, you can use a microscope, but there are cells that you can see without a microscope. In fact, you may have some in your refrigerator if you have eggs. So eggs, when they're unfertilized, are the largest single cells we have. Once an egg becomes fertilized, then it becomes more than one cell because it starts to divide, but an unfertilized egg is a single cell. So you can see here one of the largest eggs that's been on the planet recently, but you can see that this bird is extinct. This is the elephant bird. Next time you're in the classroom, take a look on the back shelf, or you can ask me to point it out. We have an ostrich egg that's been drained, but it sits on the back shelf, and it's about yay big. It's the biggest eggs that are laid today. So you can also see hummingbird eggs. They're tiny as well, but they're actually some of the bigger eggs that you can see. Fish eggs, those are also ones that you can see with your naked eye, as well as frog eggs. And these are starting to become some of the most expensive eggs in the world, including human eggs. Human eggs are thousands of dollars per egg, and this has to do with the fact that they need to be harvested. So most people don't realize that in order to be an egg donor, you have to undergo not just the surgery to harvest the egg, but you also need to undergo fertilization treatments. You have to have injections of medication in order to boost the production of eggs, because in any normal month, women only produce one egg, if you're lucky, two. And in order to do donation, you need to have them produce a lot of eggs. So they have to undergo hormone treatment in order to do that. And so they're going to be compensated well for that. All right, so we know what a cell is. We know how big they can be. Most cells are microscopic. So you can see something like a paramecium with your naked eye, but it's just going to look like a dot. So most of the cells, in order to see individually, are going to be microscopic. Now, all cells, though, do have the cell theory applied to them. And just like the theory of evolution, we have cell theory. And there's two parts to cell theory. You can see the picture down there. So the first part is that all cells are made up of one or more cells. So that's pretty straightforward, right? And that part of the theory was really easy to come to, because we could look. Once we were able to use microscopes, we could look and see, oh yeah, look at that. You know, whether it was an individual microscopic organism under the microscope, or, we looked at multicellular organisms and we could see the individual cells packed together, but one or more cells make up living organisms. The second part of the cell theory took a little longer, and that is that all cells arise from pre-existing cells. Basically, you have one cell and it divides into two new cells. And this just took longer because we had to observe it, right? Because remember, theories are hypotheses that have been tested over and over, multiple types of hypotheses all tied together. And so this one took longer just because it took some time to be able to observe this. And not just one organism, but many organisms. Now, you might be wondering, well, where did the first cell come from? That we don't know. That's still a hypothesis. So there's actually multiple hypotheses out there about the origin of the first cells. But cell theory itself is that all organisms are made up of one or more cells, and those cells arise from pre-existing cells that are here today. Now, there's some other things that we can generalize about cells, and that is every cell on Earth falls into two basic categories. So you're either one type of cell or the other type of cell. So do you know what these two names are for these cells? So you're either a blank or a blank cell. And it's okay if you didn't know, because that's part of what we're here for. So you're either going to be a eukaryotic cell, which is what we're made up of, and a eukaryotic cell has a nucleus, right? That central section of the cell that has the cell's DNA. And if you're a eukaryotic cell, you're a eukaryote. If you're made up of eukaryotic cells, like we are, we're multicellular, we're a eukaryote. So eukaryotic cells, they're going to have a nucleus. This is different for a prokaryotic cell. Pro means before. So before a nucleus, karyote means a kernel or a center. And so this does not have a nucleus. So prokaryotic cells have no nucleus, as we'll see in the diagrams for prokaryotic cells. The DNA just simply hangs out in the middle of a cell. We do commonly see it called a nucleoid region because it kind of is central, but it's not actually inside of a structure like a nucleus. And just like with the eukaryotic cells, we call prokaryotic cells prokaryotes. The thing is prokaryotic cells, they are all single cells. There are no multicellular prokaryotes. So we'll see as we go later into the semester, there are basically two groups of prokaryotes, but the ones that you're most familiar with are the bacteria. And so we're going to take a look at the basic structure of a prokaryote. We're going to start first with the structures that are found in all prokaryotes. And then we're going to look at the kind of add-on features. So almost like the bling that some of them can add. Right, so the first structure on a prokaryote that all prokaryotes have is a plasma membrane. We'll actually see that this structure also is found in eukaryotes. So this isn't just unique to prokaryotes. And so plasma membrane in this picture, it's going to be the one that's closest to the inside. And that makes sense because this is kind of like the wrapper, I almost always think of these kind of like as a Ziploc bag, and everything else is inside. Because what plasma membranes do is it keeps the contents of the prokaryote inside, right? So all of the other material that we're going to look at is inside of that plasma membrane. And in some ways, it kind of works like a Ziploc bag. It's zip closed, but some stuff can leak through it. So all of them have a plasma membrane, or you might see it listed as a cell membrane. They're all going to have this cytoplasm, and I'm never going to ask you to label or identify cytoplasm because it's this jelly or jello-like fluid that's inside of the cell. It's basically the material that fills up the cell, so it kind of helps give the cell shape. This is also something that we'll see as in eukaryotes. It's what allows material to move through the cell. So it's, like I said, it's jelly or jello-like. So it's not all water, but it's not complete solid. So it's kind of like jello that didn't quite set up. And so that is going to fill the cell. Then floating around in that cytoplasm, we're going to find DNA. And that DNA, you're going to find one very big loop of DNA, one large, what we call chromosome. That's the main DNA. But in some of them, and we'll talk about what these are for later on in a future chapter, you might find some other small circular, tiny loops of DNA. But that's another thing that all of the prokaryotes have in common, is that their DNA is circular. So it's always found in these circular loops. So we've got plasma membrane, we've got cytoplasm, we've got DNA in circular loops. And so far, all of this stuff is materials that we're going to find in eukaryotes. We're going to find these in all prokaryotes. We're going to find them in eukaryotes as well. We'll see that there's a difference on how the DNA looks, but we're still going to find DNA, the genetic material, like we found out about in the nucleic acid section. And then we've got ribosomes. Ribosomes are also found in eukaryotes. We'll see they're generally found in another structure, but in prokaryotes, the ribosomes are hanging out in the cytoplasm, just kind of floating around. They're the little tiny dots. They look kind of like pepper. These are little structures that work to take the information from the DNA and turn it into proteins. So you saw these again with the information from the nucleic acids. So in the section where they were talking about RNA, you saw the ribosomes. So these are the little factories that make the protein. So those are the structures that are found in all prokaryotes. Let's take a look at some of the add-on features that some of the prokaryotes can have. So some of them can add on a cell wall, that's the green layer here, that will protect and give them some shape, as well as a capsule. So a capsule, that's the brown section. And this is another protective outer coating. So they can add these outer layers to give them extra protection from the environment, from attackers, whatever it might be. They actually commonly are basically waging chemical warfare against each other and against other organisms like fungi in their area. So they need these kinds of protective structures against each other. Some of them, now this bacteria or prokaryote has all of the structures, but that's not common. Some of them will get these pili. So these pili are these kind of hair-like projections. Now they look like hair, but they're not hair. What they're going to do is be used to stick to stuff. So they look like hair, but they work more like Velcro. And so they're going to stick to surfaces and sometimes stick to each other to transfer DNA. So why would a prokaryote or a bacteria want to stick to a surface? Yeah, and there are lots of different reasons, right? Maybe that's where their food is. They need to infect something, right? There's lots of reasons for a prokaryote to want to stick. And then the last structure that they can add on is a flagellum. So it looks like a tail, and these are going to help them move. It's exactly like a tail because they are often going to be in some sort of fluid. whether even if it's just a little tiny amount of water or something like that. And so they can whip this around. In fact, they actually turn it. They've got a little tiny motor at the bottom of each of these. And so they can use these to move themselves around. And they, just like the pili, they don't have to have just one. In fact, I have one of my little stuffed buddies here. This is E. coli. And E. coli, as you can see, has lots of flagella. In fact, it makes him look like he's got a little beard. And so this is actually one of the ways that we can identify E. coli, because it's got tons of those little flagella. But as you can see, I'll bring him back, no pili. He's really smooth. So those are all of the structures for the prokaryotes. We'll pick up in the next lecture with the eukaryotes, because actually that's all I have on the prokaryotes. We'll see more about the prokaryotes in the microbes chapter and in the cell division chapter, but they're pretty simple, basic cells. That's all they've got going on as far as structures. Eukaryotes are much more complex.